Good afternoon, everybody. Thank you for joining us today. Before I go any further, just a quick check. Liz, can you hear me okay? Yes, thank you, Heather. So thanks for joining us today. My name is Michelle Heacock, and I'm a program officer at the National Institutes of Environmental Health Sciences, or NIHS. I'm pleased to welcome you to the Human Health Exposure Analysis Resource, or HEAR, Exposomics webinar series. This is the 10th webinar in the series. It'll focus on chemical characterization of environmental samples. So we're featuring this topic because environmental epidemiology and exposomic studies, say that 10 times fast, often rely upon measurements of blood and urine to evaluate exposures and health effects. However, analysis of environmental samples can provide complementary and unique information that can't be gleaned from biomonitoring alone. So in this webinar today, we have Drs. Heather Stapleton and Lee Ferguson, who will discuss both targeted and non-targeted approaches used in analyzing environmental samples. So this includes water, dust, soil, and personal measurements such as hand wipes and wristbands to address important questions about exposure sources and pathways. As you are getting oriented, and I'll remind you again, if you have any questions as you're listening to the presentations, just go ahead and pop them through the Q&A box, and we'll give you a chance to raise your hand during the Q&A time, but just wait till the Q&A time to do that. So now let's first welcome Dr. Heather Stapleton. She is the Ronnie Rochelle Garcia Johnson Distinguished Professor at Duke University. Dr. Stapleton will discuss chemical characterization of environmental samples. Heather, I'm going to turn this virtual podium over to you.
Great. Thanks, Michelle. Let me go to my slides here. Okay. Are they coming through okay?
They are, and I can hear you well.
Great. Thank you. Well, thank you for that very nice introduction, Michelle, and thank you for the invitation to participate in this webinar today. As Michelle just indicated, my colleague and I, Lee Ferguson and I, will be giving this talk today about chemical characterization of environmental samples. Lee and I are both part of the NIH HEAR program, and we direct the Duke HEAR lab resource. For any of you not familiar with HEAR, HEAR is an acronym for Human Health Exposure Analysis Resource. It is a centralized network of exposure analysis services, meaning it's a network of exposure science investigators and analytical experts that can provide measurements of exposure in different types of samples. So these services are available to eligible researchers who want to add or expand experimental exposure analysis into their studies of human health. So an opportunity for those of you who may have archived or banked samples to receive analytical support in measuring environmental exposures. But what do we mean about environmental exposures? Here we're primarily talking about, or the context is maybe metals, trace elements, or organic contaminants in our ambient environment. So thinking about chemicals we come into contact with, typically on a daily basis, and that may be absorbed into our body and may be a risk for certain human health outcomes. So this could be combustion byproducts in the air you breathe. It could be pesticides in the soils where children are playing. It could be PFAS, per and polyfluoroalkyl substances in the food or drinking water. Right? All of these are environmental exposures that may be of concern and which the HEAR Lab Hub can provide some assistance with. Right, so today I'm going to be talking about targeted analysis that are available through the HEAR program. Lee will talk a little bit more about non-targeted analyses, but the HEAR program can provide access to analysis for a wide range of different compounds, as you can see here, both in biological samples, primarily urine and blood. But today I wanted to talk about measurement and analysis of environmental samples for a wide range of compounds. So you can see here examples are drinking water, house dust, or even wearables like wristbands. But in selecting samples to support your research study, it really depends on your question of interest, right? So if your question is, are chemical exposures associated with an adverse health outcome? Well, then it probably makes sense to focus on the analysis of biological samples, and particularly blood or urine. And the choice of blood or urine often depends on whether an established biomarker of exposure or method has been established. Maybe it depends also on the half-life of the chemical. Typically, we use blood for chemicals that have long half-lives, urine for chemicals that have short half-lives. But we also must consider whether or not a good biomarker has been identified, a biomarker of exposure has been identified for a chemical of interest. There are some chemicals where they may have short half-lives in the body, but we don't know enough about their toxicokinetics to identify a good target analyte or a metabolite to focus on. So in those cases, analysis of environmental samples may be beneficial. Or your question might be, what is the source of this chemical exposure? Or what is the route of exposure? What pathway by which I'm exposed? So these are certainly important questions. If you want to know how to reduce your exposure, mitigate your exposure, you have to know where that chemical is coming from. So these are cases where environmental analysis of environmental samples may be beneficial. Right. So I'm going to talk today about two examples from my own research, highlighting the importance of analyzing environmental samples to answer important questions. So we're going to start with this first question: what is the primary source of exposure to brominated flame retardants in the general population? So I'm kind of going back a little bit in history and talk a little bit about polybrominated diphenyl ethers, which are now a legacy compound, or many considered to be legacy compounds. It's a persistent organic pollutant. A wide range of different chemicals are included in PBDEs, and they were historically sold under three technical commercial mixtures, commonly referred to as penta, octa, and deca BDE. The components in penta BDE in particular seem to accumulate to the highest are most abundant in human tissues and were commonly detected. But due to concerns about their persistence, their bioaccumulation potential, and toxicity, this did lead to a voluntary phase out in the US in 2005. These chemicals are now clearly established as neurodevelopmental toxicants, endocrine disruptors, and some are potentially considered carcinogenic. Right? But when we first started identifying and measuring these chemicals in human tissues, we didn't really know the source of the chemical. And there's a lot of presumption that the source was dietary. My group over the last 10 to 15 years has been really interested in understanding pathways and routes of exposures to a wide range of chemicals, including flame retardants. So to this point, we've conducted actually 3 cohort studies here in Central North Carolina, where we have a experimental design that involves visiting people's home and collecting paired samples within the home. So this includes vacuuming the home. Our research team actually vacuums the home to collect dust as a measure of what's kind of coming off products in the home. We collect personal measures of exposures, which might be hand wipes. Later we'll talk about wristbands, but also measurements of internal dose. So in this case, serum samples. In one of our first studies that we published back in 2012, we wanted to say, well, is this source really dietary, or could it be an indoor source for this exposure to this class of compounds? So by analyzing paired samples of house dust, hand wipes, and serum for PBDEs, we found that all the PBDEs in all 3 of these were significantly correlated. So what was in someone's dust was a predictor and was correlated with the levels that were measured in serum. You can see these are Spearman correlations here, 0.34, statistically significant. We observed a very similar correlation between house dust and hand wipes. So these are just measuring residues on the skin surface of the hand. But we saw the strongest correlation between measurements of what was on the hand wipes and in the serum, as you can see here. So this, to us, suggested that the source was the indoor environment. It was likely not dietary, because we could explain a lot of the variability in the blood measurements. Right, so here I'm just showing you the plot from our publication, where you can see in the x-axis is the measurement of major PBDEs that are present in serum as measured on the hand wipes. So this is just a cloth gauze with rubbing alcohol measuring the surface of the hand. And you can see how well it correlates to measurements that were made in serum. Right. But then the next question, well, what is the source in the indoor environment? What's the source to the house dust and the hand wipes? So to do this, we wanted to kind of delve further in and focus on specific products. Our hypothesis was that it was the furniture that was the primary source of exposure to penta-BDE. And here we have a focus on BDE47, which was one of the main components of that commercial mixture. Up until this point, there had been a number of efforts trying to focus in on exposure to PBDEs. But because of the difficulties in identifying source, one of the approaches used was just to use a survey and count the number of foam-containing products in the home. But unfortunately, those analyses did not prove fruitful and often did not observe a correlation. So it was unclear how much of a role the furniture played in terms of our overall exposure. So what we did is we kind of leveraged some of our resources and ended up collecting samples of products in the home. So if we go back to our study design and some of our cohorts, at least in cohorts 2 and 3 here, you can see that one was an adult cohort, one was a children's cohort. When we were in the home, we actually asked participants if they'd allow us to collect I call it a sofa biopsy. So we, if they had a zipper on the cushions of their sofas, we asked if we could open that and take a very small piece, a marble-sized piece of the cushions, because if it was treated with flame retardants, that's likely where the treatment was applied. And we could bring that back into the lab for analysis. And again, we could use this approach to look at the relationships between these different samples. So in the laboratory, we can actually take these foam samples, we kind of put them into a test tube with some organic solvents, and we use sonication to actually pull out those flame retardant chemicals from the sofa, the cushion, into the solvent, and then use mass spectrometry to screen for common flame retardant chemicals in the products. So here we often just classified the analysis of these foam samples as either yes or no. Did we detect a flame retardant, and which one did we detect? With a threshold of 0.1% by weight for the measurement of flame retardants in these products. And so here's results from one of our studies. This is from the adult cohort, but this has actually been replicated in our other cohort, where what you're seeing here on the right are basically the measurements of these PBDEs in either house dust or serum. So house dust is on the top panel, based on whether or not we positively identified this flame retardant treatment in the sofa that was present in this home. So in this case, if we're looking at the top panel, there were 18 homes, and this is in 2014 to 2016. So it was a while after the technical phase out of Penta, but there was still quite a number of homes that contained Penta in their furniture. We had 18 homes in which Penta was positively identified in the foam from the sofa. And then we had 86 homes where either Penta was not present or some other flame retardant was present. And if you categorize the dust measurements from those homes, you can see that the presence of Penta in the sofa led to a significantly higher level of this chemical detected and measured in the house dust sample. So 6.4 times higher on average, which was quite statistically significant. We observed a very similar relationship when we looked at the serum levels of BDE47 in their participants. You know, if Penta was confirmed as being present in their sofa, occupants of that home had on average 2.5 times higher levels in their blood compared to occupants in homes where Penta was not present. So again, this reinforced the notion that yes, the furniture is likely a primary source of our exposure to this compound. It's not typically diet. Certainly diet can be an exposure pathway, but maybe not as important as the furniture. And again, if we wanted to do something about this, then we need to kind of remove that source. We are continuing to work in this, and we've observed similar trends for PBDE, this relationship with PBDEs in the children's cohort. And we also are looking at some of these alternate flame retardants like Firemester 550, and again, observing a similar trend in relationship with furniture. So it's showing its importance. So now I'm going to pivot and a little bit talk about a different question when we're analyzing environmental samples. In this case, how much do our exposures to some of these chemicals change based on our activity patterns? And here I'm going to use some of the examples from our research working with the Durham County firefighters here in North Carolina, in which we use silicone wristbands to measure their exposure. So some of you may have tuned in to the HERE Exposomics webinar last month when Stephen O'Connell talked a little bit about the silicone wristband. So hopefully some of you have some knowledge on the silicone wristbands. These were first introduced by Steve O'Connell and Kim Anderson at Oregon State University in this publication from 2014. So they're very similar to wristbands that everyone wears that you receive at fundraisers. It's just polydimethylsiloxane. Studies now suggest that these measurements on wristbands can provide very good measurements, predictions for internal dose. And they can integrate exposure that occurs via multiple routes, including inhalation, dermal, and inadvertent dust ingestion. However, they do not obviously serve as a surrogate for measurements for dietary exposure. And right now they primarily work for organic chemicals, not metals. So all of the research to date is focused on exposure to organic chemicals. But what's nice about these is that you can wear these wristbands and it provides a nice measure of your average integrated exposure during the window in which you wear the wristband, right? So unlike measurements of chemicals in blood or urine, it's really hard to know when that exposure occurred. But if you're using a wristband, you know exactly when the exposure occurred, and you can look at differences in exposure profiles, maybe depending on activity patterns. Right, it is important to note that these wristbands must be cleaned first. If you buy them commercially, they will have a lot of background chemicals on them. They have to be kept in airtight containers until used. And in most of our studies, we ask participants to wear them for 5 to 7 days, just based on logistics of the study design. And we ask people to wear these continuously, when they're in bed, when they're in the shower, wear them and try to forget that you're wearing them, because we don't want people to change their behavior while they're wearing them. And so then we typically have people wear them for a period of time in which the uptake is still in the linear phase. And we can, they do not saturate for the chemicals that we're focusing on. And then it provides a good measure of exposure that these individuals are receiving. So in this case, we use wristbands to investigate firefighters' exposures. And this was a project led by my PhD student, Jessica Levasseur, in which we published just last year. In this case, we wanted to ask very specific questions about firefighters' exposures to a range of different potentially hazardous chemicals, and understand how those exposures changed off duty, maybe when they're at home, versus on duty. But further, we wanted to ask the question, how do your exposures vary depending on when you respond to a fire versus maybe then when you're spending time training or in the fire station? So what kind of a change do you experience from participating in that actual fire call? Right. And this is just an example of the exposure readouts, because we did share our findings with the research participants, the firefighters in this case. We thought this is very important. And I like this image because it actually helps helps you see some of the differences in their exposure profiles that we gleaned from using these wristbands, right? So here you're seeing data, wristband measurements for 4 different PAHs, which are outlined on the x-axis. And the symbols represent the measurements made on different wristbands depending on what period of time that was. So the house represents exposures off duty. The fire symbol, the red, represents exposures when they were working and responded to a fire. And then the brown circles represent exposure measurements while they're working, but they did not respond to a call during that 6-day period. So you can clearly see in this case, and each circle represents a different participant in the study and a different wristband sample, that while some of the firefighters were responding to fire calls, they had significantly higher exposures to some of these PAHs. You can see benzoate pyrene, for example, increased quite a bit in some of the firefighters, likely depending on how close they were to the fire, how long they spent near the fire. But we can then use this data in a regression model to look at average differences during these different activity patterns or work periods. So here you're seeing output from a regression model, a GEE model, where we're looking at differences off duty versus on duty. So each PAH is along the bottom here, and the one line that's horizontal represents your average exposure off duty. And so any symbols above that one line suggest that there was on average higher exposure to that PAH compared to off duty. And if it's a symbol that's filled, that means it was statistically significant in our model. So most of these were statistically significant because they are filled squares and circles. The squares are exposures when they are on duty and reported to a fire. Circles are when they're on duty but did not report to a fire during that 6-day period. So you can clearly see pH exposures increased while responding to a fire. But what was interesting to me is that our data suggests that exposures increased to PAHs even when they didn't respond to a fire. So something else about their activities and maybe the materials they were engaging with during that period increased their exposure to these compounds that was not necessarily a fire. And so we think this is also important information to help firefighters understand those exposures and potentially mitigate those exposures in the future. But we didn't just look at pHs, we did look at a variety of other chemicals. I'm not going to go through all the panels, but what's interesting to note here is that we saw almost an opposite trend for some of the phthalates. So here, these are all acronyms for different phthalates that were measured in the wristbands. And what you can see here are most symbols hover around that one line, suggesting that the exposures on duty were not actually significantly different from when they were off duty. And if anything, there was a few of these phthalates down here, these low molecular weight phthalates, where their exposures was higher off duty compared to on duty. Many of these are used in personal care products. So this suggests to us that firefighters are having higher exposure to these while they're off duty, probably because they're just using different personal care products when they're at home versus when they're at work. We also saw a similar trend for some of pesticides in our panel, primarily pesticides that are often used in flea and tick medicine. So again, probably just due to differences in spending more time around pets at home versus when they're working. But again, this highlights how you can use these in a manner to look at differences in exposure profiles depending on your activity. We also do support suspect screening analysis with the wristband. So it's not quite truly non-targeted, but it's comparing our mass spectral readouts to a database and trying to match and tentatively identify some compounds. So we can use this finger approach to compare responses from a mass spectrometer. Here you're looking at an example of the mass spectrum for lilial, which is a aromatic aldehyde commonly used for fragrance and personal care products and cosmetics. We do commonly see this in the wristbands as well. So we kind of use this approach to say what else is on the wristbands that firefighters might be exposed to. So we kind of, instead of trying to identify everything, we do kind of narrow down our approach and focus on chemicals features that are commonly detected in at least 50% of the wristbands. And then we kind of use volcano plots to look at differences in their exposure profiles on duty versus off duty. So here you can see in the case of the firefighter study, we had somewhere between 1,800 and 2,500 features that were identified in some of these wristbands. And so I'm going to show you a volcano plot. And in case anyone's not familiar with the volcano plot, what we're really doing is kind of looking at differences in the abundance of these signals, these features. Each feature is presumably a different chemical, and how it compares between two different conditions. And here the conditions are whether the firefighter was on duty, responding to a fire, or off duty. And so the x-axis are the differences in the abundance of that response. Your y-axis is the p-value for that statistical analysis. And this is research that's supported by Nicholas Herkert, who's a research scientist in my group. Right, so here, for example, is a volcano plot from our wristband analyses where you're looking at both of the target analytes, but some of these other features that we were able to detect using high resolution mass spectrometry. So what I first want to draw your attention to is you see the yellow vertical line in the middle. If exposures were equal, we might expect these symbols to be symmetrical on both sides. But what you can see are there are more dots, more features that were more abundant off duty than on duty because we have more of these symbols to the left of the vertical line, which was interesting. Again, suggesting that firefighters in this case have more exposure overall to things that we're picking up by GCMS off duty versus on duty. But what we're really interested in are looking at these features in the upper right-hand quadrant. These would be features, exposures that are higher in firefighters on duty when responding to a fire event and are more statistically significant, right? So again, the x-axis is the log2 fold difference in these exposures. The y-axis is the p-value. Some of these are in our target panel. Those are the ones in the red-brown color. The unknowns are in the blue. We do use an approach to tentatively identify some of these chemicals. And what you can see here is that these chemicals in the upper right-hand quadrant, many of them were combustion byproducts and PAHs. We also identified 6,2-FTMAC. That's actually a PFAS compound that is known to be present in firefighter turnout gear. BD209, which is a flame retardant. And then some of the chemicals that were identified on the left were more of the personal care products. So again, suggesting more use of personal care products at home versus on duty. But again, hopefully you can see that this provides us an ability to look at these exposure fingerprints or exposure profiles and how they differ, depending in this case on what the firefighter was doing at the time. So anyway, I know I'm kind of going over time. I'm going to wrap it up, but I'm hoping this kind of convinces you or helps you understand the importance of analyzing environmental samples. And just certainly want to advocate for thinking about linking environmental samples with measurement of biological samples. In addition to thinking about using wearables more often, particularly silicone wristbands, because I think they can help us glean insight into exposure profiles and how they change over time. Because it's a lot easier to deploy a wristband and collect it than it is to kind of collect blood samples multiple times. So anyway, I'm going to stop there and certainly thank all of my collaborators and the participants in our study, particularly the firefighters. And certainly the NIEHS for funding. And I'm going to stop there. Thank you.
Thank you for a great presentation, Heather. That was a lot to absorb, no pun intended. Just remind those who are listening, please put if you have any questions and answers, pop them in the Q&A pod now. I'm happy to read them out. If you prefer to ask it live, just raise your hand. I'll be looking for that. And Liz, if I miss somebody, you just ping me and I'll make sure I grab them. In the meantime, we had a couple of questions that came in just kind of regarding study design and considerations. So the first question is, when collecting environmental samples, should investigators focus on collection of house dust, indoor air, or perhaps using the silicone wristbands, which you just finished telling us about? And so what are the disadvantages and advantages of each sample collection?
Right. Yes. Thanks for that question. So we have to recognize that when we're collecting house dust samples in particular, or measurements of indoor air, you're sampling one microenvironment in which someone might be spending some time, right? So if your question is, what is the exposure in that one microenvironment? Again, then that's probably an important sample to collect and analyze. But if your question is overall, how much is your exposure to this chemical from multiple microenvironments, this is where the wristbands might actually be more advantageous, because it does integrate exposures from different microenvironments because it goes wherever the participant goes. In our studies where we've had paired samples of dust and wristbands and biological markers of exposure, we see the strongest correlations with internal dose with wristbands compared to house dust. So again, if your question is, what is your overall exposure? I think wristbands are better. But if the question is, what is the exposure in the home? Then I think that's where it may be more important to focus on house dust. If it's a chemical more likely to be present in dust, and that's a function of its physical chemical properties, or if it's volatile enough, we might want to sample air.
A question building on your comment on the wristbands is, are the wristbands available for clinical medicine? And what is the cost, and who provides the analysis?
Another good question. So the wristbands are certainly available for use in many different applications, certainly within a clinical, although I don't believe there's any clinical labs that are measuring these. Mainly it's academic researchers that are analyzing wristbands at this time. Kim Anderson's group does have the My Exposome company that provides wristbands commercially, so people can look that up online. I'm not sure of the costs. But there are a number of academic researchers that are collaborating on large studies. For example, I'm collaborating with two groups that are supporting some cancer cohort studies, in which we're providing the wristbands to these researchers to hand out to the participants, and those will be analyzed at a later time. So I think there's multiple opportunities to get access to using wristbands at this time. And as I said earlier, the HERE program does provide support for analysis of wristbands.
Thank you, Heather. Okay, so there's some other questions about these wristbands. So another one is looking at it from a different direction, kind of the end of the line. You have the results, and we're thinking about reporting these results back to participants. So how do you report, how do the report backs for participants look like for data obtained from the wristbands, given this novel method? And how is it translated in a way that's understood by the participant receiving information?
Right. So yes, we do always try to use a little caution when we're sharing the wristband data, because we can't translate a wristband measurement into a blood level, right? So we have to make that clear. But actually, one of the slides in my presentation was actually a snapshot of the report out we used with the firefighters here in Durham County. So we kind of use a strip chart approach where we show the measurement of the chemical on the wristband for all the participants in the study, and then we identify which wristband measurement was theirs. So they have a sense of what their exposure is to those chemicals relative to the study population. And then we try to discuss what are the sources of the exposure, what do we know about health risks for this chemical, what don't we know, and what are steps we can do to mitigate our exposure. And so that's the context in which we've been providing data or report backs on the use of wristbands to this point.
Thank you, Heather. There are a couple more questions, but I'm going to save them. Audience, there is a time at the end of the presentations just for questions for both Lee and Heather, should we have the time. And if not, Heather, would it be something that it would be okay that we could get that question, that answer to participants who are asking it by email, say?
Of course,
Okay.
happy to answer.
All right, excellent. Okay, we are going to now turn this over to Dr. Lee Ferguson. He is also from Duke. He is the Associate Professor of Civil and Environmental Engineering. He is going to present on non-targeted analysis for novel compound discovery and environmental samples. Dr. Ferguson, the virtual podium is yours.
Great. Thanks, Michelle. Can you hear me okay?
I can, and I see your slides as well.
Wonderful. Okay. Yeah, so I'm going to talk a little bit about some complementary work, the other side of our HERE Analytical Center here at Duke University from what Heather discussed. My laboratory focuses primarily on non-targeted analysis, and we use this in a variety of ways, mostly for compound discovery, and we're applying it to a number of different environmental samples. My laboratory tends to focus mostly on water and exposures that are relevant to drinking water situations. Okay, so right. So as an environmental chemist, I spend an awful lot of time thinking about this question, which is, which organic pollutants occur in environmental waters and what are the implications of that occurrence? There's a lot of challenges around answering this question. And so applying non-targeted analysis to try to answer these kinds of questions has been something that our community has been involved with for about the last 10 years. And so it's been interesting to watch how that's evolved. The challenges, though, still remain. One of the biggest is the vastness of chemical space. And so, one of the big issues that we face is how do we decide which chemicals in commerce are really important when we think about emerging contaminants. And so down in sort of the bottom of the slide, you can see actually projection of chemical space from a variety of different databases. So the gray dots are basically 65 million molecules that were present in PubChem when I pulled this down back in about 2019. And then if we overlay that with a number of different databases like the EPA's DSS Tox, their chemistry dashboard, the Office of Pesticide Programs, the TSCA database. And you can see that all of these different collections of molecules that we might think of as environmental contaminants occupy different areas in the chemical space projection, depending on how we visualize this according to molecular descriptor. And not only do we face issues and challenges with accessing all of these compounds analytically, but also we face challenges based on just the sheer volume of compounds that we're trying to annotate. So unfortunately, when we try to do research on chemical use and production, these lists and the compilations of chemical use is just not comprehensive enough for us to really prioritize. And then, as I'll talk about here in a few minutes, transformation in the environment, biotransformation, either in aquatic environments or during wastewater treatment, for example, really takes this already complex set of molecules and makes it much, much more complex. And so this is the real challenge that we face, and we put that problem statement out there. And so then always the question has been, how do we address this? And so this is actually one of my very favorite scientific cartoons back from when I was a graduate student in 2001, and proteomics and genomics were really starting to come into their own. And it became clear that we could use high performance analytical chemistry methods to access chemical complexity in a biological system, such as a human being. And so I started to ask the question, well, why can't we use some of the same technologies to do environmental chemistry? And really that's, I think, where we've come to. We've stopped fishing for individual molecules one at a time, and we've started doing what this fellow over here on the right is doing, and really trying to look at our environment as a system. And so the real limiting technology there is our analytical chemistry. Okay, so I don't want to sort of talk a lot about the instrumentation that we use, but I will say that the linchpin for doing non-targeted analysis of environmental contaminants is also very much the same linchpin for doing metabolomics assessment in exposomic studies. And this is ultra-high resolution mass spectrometry. And so this is instrumentation. Here's an example. This is an Orbitrap mass spec that we use in our laboratory. And it really allows us to access high resolution and high mass accuracy measurements of contaminants, and also get structural information due to the fragmentation and fragment spectra that we're able to generate. And so we use these technologies as primary tools in our non-targeted analysis. So I'm going to go through two examples for you of how we use these technologies in our laboratory to do environmental contaminant assessment relevant to human exposure. The first one is non-targeted analysis of PFAS in North Carolina waters. Okay, and so I probably don't need to spend a lot of time introducing PFAS. We all have heard of these compounds. And what's interesting about them is they really span the gamut from compounds like C8 and PFOS over here on the right that are considered legacy PFAS. These are compounds that have been used for a long time for things like, grease-resistant coatings, firefighting foams, stain repellents, etc. And then also emerging or next generation PFAS, such as the Gen X molecule here over here on the left, that are in some cases subtle and in some cases rather drastic changes to the chemical structure of PFAS, but they all share this high fluorine to carbon ratio and a rather remarkable persistence in the environment. This has made them, of course, of concern for biological exposure. PFAS production over the years. Well, here's the timeline, or a timeline. Really starts back in the 1940s and into the 1950s with patents from 3M, DuPont, and then proceeds through the middle of the 1970s as alternative chemistries such as fluorotelomers have been produced. And then different changes in the market and changes that begin to emerge in the early 2000s due to regulations and due to the recognition that these compounds may cause concern due to exposure. Most recently, things like 70 part per trillion health advisory level that was instituted for drinking water back in 2016. And then more recently, I should update here in 2023, the new draft maximum contaminant levels issued by the EPA for drinking water that are hopefully going to come into enforcement pretty soon. And all of this development has led to increasing attention to these compounds in human exposure. It's made much, much more complex by the alphabet soup that we find ourselves in when we are analyzing PFAS. Okay, so this is a paper that was published by Zheng Wang and some colleagues that really attempts to lay out PFAS into chemical families. And what you can see over here on the right is the real complexity of how PFAS are made and used. All of them share sort of this basic structure of high fluorine to carbon ratio, but with different sets of functional groups that make them suitable for different uses in commerce. It also leads to increasing complexity, both for environmental fate, possible transformations, and ultimately analytical chemistry, how we measure these compounds in the environment. We have everything from our legacy PFAS up at the top here, which have thousands in some cases of peer-reviewed articles since 2002, all the way down to some of our emerging compounds like our ether acids here, like Gen X, et cetera, that are really underrepresented in the literature and we don't understand as much about them. So if we put all these together, there's some conclusions we can make. You know, these are compounds that have high chemical stability. They're unique with respect to their partitioning behavior, and there's a lot of them. I mean, we're talking somewhere around 3,000 to 4,000 compounds in our mix. So how do we start to access these analytically? Well, in our laboratory, what we've set out to do is come up with a comprehensive and systematic analytical pipeline for PFAS annotation using high-res mass spec. So we start with lists of compounds, and we're able to use both the EPA's chemistry dashboard and the compilations of PFAS chemicals that they have created, as well as some in-house databases based on analysis, careful, systematic analysis of things like firefighting foams, to come up with lists of compounds that might be present in consumer products or potentially in the environment. We can take those lists of compounds and use cheminformatic approaches, such as biotransformation predictions or the EPA's chemical transformation simulator, to simulate and predict biotransformation products of these PFAS that are present in commerce. Okay, so now we're mimicking what might be happening in water treatment and wastewater treatment or in biological systems. To make these molecules then accessible for our mass spectrometer, we can take all of these compounds and use some in silico MSMS prediction algorithms to actually produce a in silico electronic spectral library that can then be searched using high resolution mass spectrometry. And once we do that, we generate a database that contains about 40,000 chemical structures for PFAS and their predicted transformation products, covering about 17,000 formulas. And what you can see from this inset diagram is that, or upset diagram, sorry, is most of these turn out to be biotransformation products, but we still have, somewhere between 5,000 and 6,000 compounds that are predicted to be in commerce or in firefighting foams. So what's unique about this database is that it actually maintains a network of chemical connections due to the transformations that were predicted using our biotransformer and chemical transformation predictions. And what you can see here is actually a molecular network centered on PFOA right here in the center. So this shows all of the molecules in our database that have biotransformations or chemical transformations that are predicted to ultimately form PFOA. And you can see that some of these are what we think of as PFAS precursors. They may be dimers, they may be fluorotelomers, all of which can transform under predicted conditions to form this PFOA. And we can go through this database and find these connections and maintain those connections when we do our analysis. And all of these databases, by the way, are available in a publication that we put out about a year or two ago. That's not the only thing that we have to worry about. So obviously the informatics are a big part of non-targeted analysis for PFAS, but also the analytical chemistry, the actual measurement of these compounds is challenging. So what we've done is come up with an online solid phase extraction method that allows us to do very fast, very sensitive, and very highly selective analysis of PFAS from environmental samples. This helps us to minimize sample handling and maximizes our sensitivity. So here we're loading 10 milliliters of a water sample onto a solid phase extraction cartridge automatically using some pumps and valves. We then vac elute that extract onto our analytical column using some clever plumbing here to elute and mix mix out the organic solvent. And then we switch and do a traditional LC separation on our C18 column. All of this is automated and done using the software. Sorry, excuse me, I'm getting over cold. With our mass spec and our LC, and we're able to do this full non-targeted analysis on a 10 mil unprepared sample in less than 30 minutes. So it performs quite well. We've actually tested it, even though we use this method primarily for non-targeted analysis. We can also do quantitation. And so we've tested, this is an example of a 5 part per trillion PFAS standard mix using this method. And you can see we're way above our detection limits. Typically, we're down in the tens to hundreds of parts per quadrillion for our sensitivity using this method. Okay, so here's an example of how we use this. We had an example where there was a AFFF release due to a firefighting incident. This is a fuel truck that actually had an accident upstream of a tributary to a drinking water reservoir. There was a lot of firefighting foam sprayed here, as you can see. Large release to a nearby creek that then emptied into the Catawba River upstream of the Belmont drinking water plant. So we went and sampled surface water samples at the spill site and downstream for PFAS and used our non-target method for that. What we found is that of the about 1,400 compounds that we're able to detect in our analysis, 92 of those were tentatively identified as PFAS. And so what we can do is actually use our molecular network analysis to find out some things about what's happening to these samples or these PFAS in the samples. So for example, if we look at the distribution of molecular weight versus retention time, we can see that in some cases, we see a decrease in retention time. You see these arrows represent basically transformations. These are biotransformations in red. And you can see that basically, as you'd expect, biotransformation produces more polar compounds oftentimes, which have lower retention times. And we see that here. In our Van Krevelen diagram, you can see actually arrows pointing to the right in many cases here. Those represent incorporation of oxygen, high oxygen to carbon ratios, again, indicating potential transformations and oxidations that might be happening in the environment as these PFAS are released into these surface waters. And so we can keep track of all these connections during our annotations. We also were able to assess the performance of our in silico MSMS predictions. And so here's examples of compounds that were annotated using our approach. These are actually the fragmentation spectra of all of these various different compounds. Cases where you see purple lines, these are annotated fragments that match both our predicted MSMS fragments from our CFMID machine learning model, as well as annotations based on a formula prediction algorithm called Sirius. And so you can see that this actually performs quite well for compounds that have extensive non-fluorinated side chains, PFAS precursors, for example. Where it starts to break down is, for example, here in these ethane thiols and sulfonic acids, where some of these red peaks don't match the fragments. And that's not necessarily surprising because fragmentation of perfluorochemicals is often poorly predicted by our in silico algorithms. So what did we end up with when we put all this together? Our molecular network actually lets us see some transformation networks between compounds that highlight some of the abundant molecules that were present and in the AFFF and released into the water. Some of these are compounds that we recognize and know about, 6,2-fluorotelomere sulfonate and some of the precursors, which turn out to be abundant components of this particular AFFF mixture. But we also found some novel compounds. All of these pink stars are PFAS compounds that are predicted in our transformation or in our databases, but have never been observed in the environment. And then we're able to maintain all of the connections and maintain which biotransformation pathways actually led to these products. Okay, so just switching gears really quickly, I want to talk about our second example. This is actually analysis of drinking water contaminants in an international study. This is in Sri Lanka, where we actually went over and took drinking water samples in communities that are impacted and are experiencing CKDU, which is chronic kidney disease of unknown etiology. And this is a real mystery where it's unclear what the risk factors and the potential exposures that may induce or contribute to the development of this disease in these populations. And so we took water from areas that do not have endemic CKDU, and then we took drinking water samples from areas that are experiencing a high level of CKDU disease. We prepped these samples in Sri Lanka and brought them back to the United States for analysis. Interestingly, so here our pipeline is a little different from what I showed before. We're using commercial data processing software, Compound Discover from Thermo, as well as some custom written scripts and algorithms that we use to consolidate and reannotate these our compound identifications. We're using some in silico MSMS prediction and annotation tools, as I mentioned before. And then because we're interested in finding compounds that might contribute to nephrotoxicity, perhaps contributing to this chronic kidney disease progression, we're using an in silico model that was published by Shi et al. In Frontiers of Pharmacology a couple of years ago that allows us to predict nephrotoxicity as a consensus binary true-false model just with structures, chemical structures as inputs. So as we annotate our molecules using our annotation strategy here with structures, we can then feed those into the nephrotoxicity prediction model to try to get an idea of potential toxicity. So we had thousands of compounds detected in these samples, but again, this highlights the chemical complexity and one of the real problems we face with chemical dark matter in environmental non-targeted analysis. Only a small portion of the compounds gave either confirmed IDs, we had 28 confirmed IDs with authentic standards, or 400 probable annotations using molecular structure, or sorry, spectral libraries in our annotation spectral libraries. We had about 3,000 tentative annotations using our in silico MSMS algorithms. Most of the confirmed compound annotations, these are our examples of compounds that we're able to confirm using authentic standards. These indicate pollution from both agrochemicals as well as wastewater. This is not surprising given the activities that are happening around these drinking water wells in Sri Lanka. There was a lot of farming, a lot of agriculture, things like fipronil, fungicides like hydroxychlorothalonil, as well as pharmaceuticals and personal care products and other components like sucralose here, for example, which are indicators of wastewater discharge. this artificial sweetener, for example. Some of these compounds, some of the pesticides in particular, and agrochemicals were actually predicted to be nephrotoxic based on our model. And so you can see that we had a real range of detection frequency in the non-endemic and endemic wells. In some cases, very similar ranges of concentrations were measured. And in other cases, we actually had higher concentrations, relatively higher concentrations in the endemic relative to the non-endemic wells. We're able to quantify all of these compounds, of course, because we have authentic standards for them. Also, we were able to find a number of polymeric series. You can kind of pick those out pretty easily visually when you look at the molecular weight to retention time distribution in our non-targeted analysis. These are things like polyethylene glycol-based surfactants. Not surprising that they would be present in especially poorly treated wastewater or in stormwater runoff in some of these samples. So the agrochemicals, many of the agrochemicals that we detected and had either probable or confirmed IDs were higher intensity. The green boxes here, these were significantly higher abundance based on intensity or concentration in the CKDU endemic wells relative to the non-endemic wells, also suggesting a link between agricultural activities and progression of this disease. And similar to what Heather Stapleton showed, we can actually look at these non-target data using volcano plots. So as she said before, a volcano plot, just to orient you, the origin here is 0, 0. You know, on the left side and right side, these are fold change relative to the other treatment. So here, everything to the right of our red line here is significantly higher in endemic wells, and it's above basically in this upper right-hand quadrant. Everything in the upper left-hand quadrant was significantly higher abundance in our non-CKDU endemic wells. What you can see is you have a significant right bias in these samples. And then we've color-coded these according to annotation and nephrotoxicity prediction, especially in the area here in where we have compounds that were higher in endemic wells. So what you see is that we have a few compounds that were either confirmed or probable IDs, tentative or probable IDs, structure annotations, which are predicted to be nephrotoxic, that are higher, significantly higher abundance in the endemic wells than in the non-endemic wells. We also have a lot of compounds here which are non-nephrotoxic, and we have a lot of compounds in these gray crosses that we still don't have annotation. We have no structure annotation other than molecular formula. And this really highlights the need to increase our annotation success with expanded spectral libraries. And then now at this point, we're actually taking some of these annotations, some of these compound annotations, and working with toxicologists to test these for potential nephrotoxicity in vitro models to try to provide insight into whether these compounds might actually be responsible for some of the progression of this disease. Okay, so with that, I'll just wrap up. So I hope I've shown you that non-targeted analysis using high-res MS, this is a really important tool when we're doing compound discovery in context of exposure assessment in the environment. Our novel PFAS discovery is really supported using both cheminformatics and advanced sample preparation strategies. So these two technologies really go hand in hand when we're trying to increase our annotation success for PFAS. In our Sri Lankan study, the analysis of these drinking water samples really suggests that agrochemicals as well as wastewater are some of the important contributors to chemical pollution, or at least organic contaminant pollution in these Sri Lankan wells. And then we need to pay attention to some of these compounds, especially that might be predicted to be nephrotoxic in these Sri Lankan well waters. And so more study is needed on that. So with that, I want to thank you for your attention. I'll thank all of our collaborators, the students who did this work, particularly Gordon Getzinger and Jake Ulrich, who are former students of mine, and then the support of HERE and other funding sources. And I'm happy to take any questions.
Excellent. Thank you so much, Lee. I have to say this hour is not near enough time to go through all the data that both you and Heather have shown us. I'll remind folks who are joining us virtually, just please, if you have any questions, pop them through the Q&A pod, or feel free to raise your hand if you prefer to ask it in person, and I'll be on the lookout to see if I see any hands raised. In the meantime, Lee, I have a couple of questions for you. So regarding your Sri Lankan story, your last vignette from Sri Lanka, and you're looking at those areas that were more endemic for CKDU, is it the case where there was a high percentage of people with CKDU, or is it the case where maybe 20% of the population ended up having chronic kidney disease?
Yeah, so that's a good question, Michelle. I actually don't know what the percentages are exactly. I'd have to ask my colleague, Michelle. I do know that what we are considering endemic, when we say endemic, it is a high fraction, elevated fraction of the population that is experiencing CKDU. In the non-endemic region, it's either not present, that is, there were no examples, or there were very, very low levels of CKDU incidence. And I should note that there certainly are geographic sort of patterns for this, and some of it seems to overlap with some of the agricultural areas, which is, of course, one of the hypotheses that we had in terms of contributing factors. However, what's also interesting is there are villages and other communities in the geographic regions that typically have high CKDU occurrence that don't have endemic CKDU within the villages. So there's micro heterogeneity, and trying to tease all that out is something that our colleagues are really trying to get at, both from an epidemiological standpoint as well as from sort of a chemical analysis standpoint.
I really liked how you were talking a lot about plugging in other ways to analyze the data and interpret the data, and to get be a little bit more robust in your analysis. Along that line, I'm wondering, so you looked for the CKDU, you looked at nephrotoxicity. Is there also a way, based on the approach, that if somebody is interested to understand if there's genetic susceptibility, could there be a way, if you've got individual genotypes, where you could understand whether or not there's an underlying maybe polymorphism that is making one person more susceptible to another? And I do apologize, this is a stretch, so you don't have to answer. You just had my curiosity piqued.
I actually think that's a great question. I wish I could answer it. I think that's actually certainly something that my colleague Nishad Jaisandara, who's my co-investigator on this, he actually should be on this slide, would be better prepared to answer. But I do think that there are underlying sort of genetic factors that have been explored. What I don't know is whether there's underlying genetic factors that make people more or less susceptible to particular chemicals that might be present in these drinking waters. And I think that'd be a very interesting question. I think that there'd be some interesting epidemiology that would be needed to tease all that out. But certainly I think that it's possible.
Thank you, Lee. There's a couple questions that came through the chat. One is, are there plans to look at seasonality changes of exposure in Sri Lanka? You could see a difference in the agricultural chemical loads or seasonal applications that might be impactful. And I would even add, heat, temperature.
Sure. Yeah, that's a great question. It is actually something we are looking at. So one of the things we're limited by is the ability to actually go back and actually grab samples. It's a long way to go, and the sampling is actually pretty difficult. But we do have colleagues in Sri Lanka that we're working with on this, and so we will be getting more samples that will be collected during different seasons. So these samples were collected, I believe, in the summertime, and so I'm not sure, when the next sampling is going to happen. It's in the next year, but certainly we're interested in seasonality. What's also interesting is agrochemical application rates change over years. Certain agrochemicals actually have been banned, but still actually are applied sometimes due to black market acquisition of some of these agrochemicals. And then there are examples where there are compounds that and agrochemicals that have just started to be used in one time period, and then they actually just kind of disappear from use. So I think that it's sort of a moving target with respect to the chemicals that are used in agriculture there in Sri Lanka and really anywhere.
Thank you, Lee. And I'm looking at the time, and we are at time. I just have a couple more things to wrap up. But before I do that, Lee and Heather, thank you so much for taking the time to get your slides together, present them today to us, and answer our questions. And Lee, especially for doing that stretch with me in some of those questions.
Sure.
So next, I just wanted to remind you all that we are taking a little bit of a hiatus over the summer, where we will be gearing up, and I think that next slide should be up. Yes. So the next topic of the Here Exposomics webinar series is clinical translations of exposomics for precision medicine. And I bet they're gonna get that wristband question, Heather. What do you think about that? And that is to be determined, but be ready. Check out the Here website. That's planned for fall of 2023. So y'all, thanks again for your time, Heather and Lee and all of you joining us virtually. And I will wish you a great rest of your day and great rest of your summer for those who are just joining us for the Exposomics webinar series.